Choosing the wrong cell motility assay can produce clean data that answer the wrong biological question. An uncoated Transwell may measure basic movement, but it cannot independently demonstrate barrier traversal. A chemotaxis setup may record migration, yet without a credible gradient it cannot establish directionality. The correct buying decision begins with the disease mechanism and the program decision the study must support. Migration, chemotaxis, and invasion are related but distinct endpoints. Migration measures movement in a selected model. Chemotaxis examines directed movement along a chemical gradient. Invasion adds an extracellular-matrix-like barrier that cells must traverse. Although these formats may use similar instrumentation, they require different model logic, controls, and interpretation. In oncology, migration can support early phenotypic comparisons, concentration range assessment, or rank ordering of candidates. A positive result means that a perturbation altered movement under the stated conditions. It does not establish tissue invasion or prevention of metastasis. When the hypothesis specifically involves passage through an extracellular matrix, an invasion format becomes more relevant. Matrix composition, thickness, preparation, and consistency must be documented because each can change the difficulty of traversal. Cytotoxicity, proliferation, and endpoint timing also require close control. Reduced viable cell numbers may appear as reduced motility, while extended incubation can combine movement with cell growth. In immunology, chemotaxis is most informative when tied to a defined receptor-ligand relationship. The design should identify the cell population, chemoattractant, expected receptor or pathway, and compartment in which the signal is placed. Conditions that test gradient dependence are essential for separating directed recruitment from nonspecific movement caused by differences in media. Primary immune cells may provide greater biological relevance, but donor variability, activation state, and limited material can complicate execution. Cell lines can improve consistency and throughput, provided their receptor expression and functional response are verified. In either case, a concentration-dependent reduction in movement should not be described as selective chemotaxis inhibition when it tracks with reduced viability. Inflammation studies face a broader translation challenge. Migration or chemotaxis assays can determine whether a disease-associated mediator changes movement in a selected immune or stromal cell model. However, they do not recreate vascular flow, tissue architecture, multiple interacting cell types, or evolving cytokine networks. A reduction in one controlled movement endpoint, therefore, does not prove anti-inflammatory efficacy. Across all three settings, application fit depends on 5 considerations: alignment with the disease mechanism, relevance of the cell model, linkage to the chemokine or molecular target, the need for a barrier, and the required throughput. Their priority changes with the question. Barrier relevance may be central to one oncology program and unnecessary in another. Ligand linkage may define an immune recruitment study but be secondary in a general motility screen. Decision-ready studies also require protocol traceability. Reports should identify cell source, passage, or donor information where relevant, seeding density, membrane and pore size, matrix conditions, medium or chemoattractant composition, compound concentrations, incubation time, readout, controls, replicates, and normalization. Each variable affects what the result can support. Controls form part of the analytical argument. Vehicle conditions establish baseline behavior. Positive and system controls confirm assay responsiveness. Negative and no gradient conditions clarify background and directionality. Viability checks address a major confounder, while replicate-level outputs expose variability. Predefined rules should govern technical failures, image artifacts, and unexpected wells. A defensible study plan follows 6 steps. Define the disease hypothesis and program decision. Select migration, chemotaxis, or invasion. Choose biologically relevant cell and signal models. Specify controls before execution. Establish replicate, normalization, statistical and data expectations, and define the next validation step. The most elaborate assay is not automatically the most useful. The strongest choice is the format that directly tests the current mechanism, produces traceable evidence, and keeps every conclusion within the boundaries of what was actually measured.